Mic's on.
Oh.
All right, thanks everyone for coming over to watch the Chalk Talk. Hopefully you've all met with Karli and seen the presentation either in person. There is a recording which didn't turn out too well, quite honestly, but you can at least hear her and see her slides. And I'm working with DLT to do a better job on the next ones. But again, we've had a chance to hear Karli in lots of different contexts, and as usual, we want to hear what her research plans are and where she intends to go with her science. So no further ado, Karli.
Great. Thank you. Yeah, it's been really fun interacting with all of you. And I'm really excited to talk about what I want to do moving forward because my research talk was more on kind of what I've done and how I've gotten here. And kind of on that, I guess I wanted to just retouch on my journey in cardiovascular research. So I got my bachelor's in applied mathematics and bioengineering here at the University of Utah, also my master's. I did my PhD in Graz, Austria, and that focused on the personalization of cardiac models. And that was based on clinical data. I moved on and I started my postdoc in the same lab. I'm doing more direction on clinical applications and also on more efficient modeling workflows, which I'll get into. And most recently, I started as an Esprit program fellow doing sex-specific modeling of cardiac digital twins, which is one of my main research areas that I'm really excited to. But I'll get into those. Hi. So on this, my primary research interest, I kind of went this on the fact that I hoped that most of you were familiar with my modeling in general and kind of what I'm doing. So just a little recap. I generate computer models of how the heart functions. And because of that, there's a lot of different research directions that I have. So developing workflows and tools. Obviously, if you're doing any sort of modeling, there's going to be a lot of infrastructure involved in terms of software, modeling, how do we even set up the models, different tools, understanding disease mechanisms and clinical applications. And then for me, and I think what really sets me apart from other people in the field doing what I'm doing is, I call it inclusive modeling practices, but essentially what this means is addressing the bias in cardiovascular research in terms of men and women. So most of these models are set up with very generic electrophysiology. There's not a lot of effort at all made in the field to account for sex-specific differences in ancestral backgrounds. And I'll move into, I guess, the first one, if there's no kind of questions.
Yeah, I should have said that at the beginning.
Please jump in at any point.
Sorry for questions.
Yes. Yeah.
We are recording this for people who can't be here. It's not being broadcast.
Mm-hmm.
You can file somewhere. These microphones work great as long as you turn them on.
Mm-hmm.
Turn them on, there's a little gray button on the bottom. You
Okay.
know they're on, and then they'll green light comes on. Just get reasonably close to them, then they work fine and they'll be recorded properly.
Okay. Yep. So going back to direction number one, I thought I'd kind of show what those points are. So developing workflows and tools. This is directly taken from my research talk on Friday. And so what a lot of those tools and workflows mean for me is this entire pipeline. So this is the over slide of generating a cardiac model. So you start from clinical data, you have to get a specific anatomy that's derived from the patient data, and then we have to tune the electrophysiology and do parameter updates. So I have developed tools and I've developed this entire pipeline. So I published this paper. It's one of the first end-to-end papers for doing modeling workflows, and it has clinically viable aspect. And a lot of those tools include cardiac segmentation. I know many of you have done segmentation, image processing. Functionalization is a bunch of tools. And then also simulation framework, so doing fast, efficient simulations. So this is kind of my first direction and research aim because there's a lot of workflows and tools that haven't been developed. The second is understanding disease mechanisms and various clinical applications. So to date, we've taken these models and I've extended it to a variety of different cardiovascular diseases. And these are a bunch of cardiac arrhythmias. And you can see here, this is kind of the cohort of all these different types of diseases that I've modeled. And I keep adding on new ones every time a cardiologist comes to me and says, I want to understand this cardiac application. I want to better understand the mechanisms. Can we use this tool to derive better clinical outcomes and help with patient care? And so that's where this direction comes from. It's really getting into the clinic, understanding how we can use these models in a clinical setting. How can we use it to derive disease mechanisms which help benefit cardiovascular care? So this kind of direction is more based on a lot of my work of working with cardiologists and radiologists and getting it into the clinic. So direction, I guess, direction number 3. So direction number 3, this is the one that's most close and dear to my heart, and the one I'm most excited about is inclusive modeling practices. So cardiovascular disease has different ways that it manifests in females and in males and across different ethnic or ancestral backgrounds. And this goes into a bunch of cardiovascular factors, which include sex, ethnic background, age, other types of compounding conditions or hormones. And all of these are not accounted for in clinical guidelines or care at this point. So they have just very generic protocols about how they decide to do treatment, how they decide to do diagnostics. And this goes directly into a lot of people not receiving treatment when they should or receiving non-optimal treatment. And what my goal is to use these models and parameterize them to start with females and males, and then move on to having more inclusive modeling, which has not been done in the field at all. So all of the models are very generic. There's only been a very few, I think 1 or 2 studies that have tried to address this in any way, shape, or form in terms of cardiac modeling. So those are kind of my 3 overarch directions that I'm working on, and these feed into more, I would say, general research interests that come together. So I will start with the first one. The color is really bad, but reproducibility and standardization. So we've developed all these modeling workflows. So right now in the field, we have a big need for reproducibility and standardization in our workflows, because right now all these different groups are popping up doing cardiac modeling, cardiac electrophysiology, but there's no standardization in the workflows, and that makes it really hard to work together, but also in terms of actually verifying that models are physiological and they're accurate, because a lot of times when you set up models, you can get simulation artifacts which actually look like clinical disorders. You can get a lot of problems with how do you parameterize these models. And this has been coming up more and more in the field, where we've started to look back on modeling studies and realize that a lot of simulation artifacts have actually led to meaningful results, and a lot of groups will reach out to us on modeling workflows. So I've started on various aspects. You can't see the titles, but we have one for reading clinical data. We have one for setting up the electrophysiology in the models. I'm working on EP simulation frameworks, and then also how do we conduct studies that are actually meaningful and clinically applicable. Oh yeah, here's all the different titles. So the next one is parameterization and personalization of models. This is also something I've kind of made a name for of myself in this field of how do we parameterize these models. So first of all, how do we just set up the parameters that go into this model? So what are they? How do we make them so they match electrophysiology? And then also, how do we personalize this for both a given patient? So how do I take a model and make it look like one of you? So say one of you has a cardiovascular disease. How do I make this model not only look like you, but behave like you? And then also, and applying this also to populations. So this is a very big research interest I have that is always, I think will always be needed and necessary. And then moving on to uncertainty quantification. So as you can imagine, because these models have a lot of parameters, getting into uncertainty quantification is really needed. How do we understand kind of the domains of what the models are applicable in? How do we decide when we put parameters into the model, we get outputs? How to, understanding the relationship between all these different parameters, which parameters are needed, which are not. So what is actually needed to generate a physiological model and what is not. I've done a lot of work on uncertainty quantification, and I plan to continue moving forward because this is really applicable for machine learning and other aspects. And then also clinical and experimental validation. So we have this model and we set it up and we say, okay, I have a model that, like, let's say looks like Chuck and it behaves like Chuck, but does it actually behave like Chuck? Does it actually do what it's supposed to? And can it actually make reliable clinical predictions? And that's where validation comes in against both clinical data and also experimental data. So I have a lot of work actually with Rob and reusing his data sets that he's done before, and then also doing different types of experimental validation. So experimental replicas, clinical Turing test, and also clinical comparison. The clinical Turing test is something that I came up with during my PhD. That's really fun. We generate a whole bunch of simulated signals and we mix it together with real signals like ECGs, and we give it to cardiologists and we say, is it real or simulated? And is it, like, what disease is it? So we can test how good cardiologists are at actually doing ECG diagnostics. I'll say, not all great. Machine learning will do really great areas in this domain, I think, and it gives us a better idea of if our model's performing like it should. And then the last is machine learning and data-driven approaches. So machine learning and data-driven approaches have started to become present in this field, predominantly in image segmentation, as you see here. But I definitely want to expand it in terms of surrogate modeling, synthetic ECG databases, and trying to use machine learning when it's really applicable and infusing that into models when modeling is more necessary. So I don't think machine learning always needs to go. And it's not always applicable, but the two can play really well together. And there's not been as much leveraging of this in the field as I would have hoped and seen. And I really want to get on that bandwagon. So yes. So those are the main overviews. Oh yeah. Okay. Is there any questions at this point? So I know I'm just kind of going through, but yes.
Please describe, how would you expect the experimental validation part? There isn't much.
Yeah.
If you could just expand a little bit on the experimental validation, what does it look like? And experimental validation of these kind of models.
Yeah, so what that essentially looks like is that someone will go and develop an animal model. And for example, here at the CBRTI in Robbie Ranjan and also Derek Drosdel does a lot of these experiments they take an animal model and they say, we're going to experiment how the heart functions. So they'll go and there's different techniques, but they'll place electrodes all on the surface of the heart to measure what is happening electrically on the surface of the heart. They'll put electrode needles into the heart itself in this animal model. And then they'll also run, so once all the needles in and they can record a bunch of data about what is happening electrically within the dog or within the pig, then they'll do interventions. So they'll first have an animal model which is just running in normal sinus rhythm, and then they'll do, like here in at the Ski Institute, they'll do interventions with degrees of ischemia. So they'll make the heart ischemic by contracting off blood flow. And through this, because it's both time progression and also like a base setting, we can use that to see if we take this and we replicate it in a simulation. So we need to parameterize it, make it replicate the animal model, and then see if we can replicate all of these different disease conditions and how the disease progresses through time. So that's the technique. So you really just generate a simulation replica of this experimental setup, which has a lot of invasive data, which you can't do in humans. And then we can compare it. Yeah.
So how transferable is it to humans? I mean, I realize that there are restrictions, right? So do you get close to any kind of data from human or?
Yeah, there's a lot of groups. So we work with a group that's in Bordeaux, France, at Lyrique, and they will actually get, so if you have heart transplants, you can think about if, someone is brain dead and they'll start to do organ transplants. Usually by the time that all the other organs are donated, the heart will no longer be viable, or it won't be viable if they have certain diseases like hepatitis B. And so what happens at that point is these hospitals will donate hearts to science if they have donated that their organ is capable of being donated. And then we'll run actual human experiments. So with actual human hearts, and we'll do the same procedure. And it's fun, but it's very rare. So I think there's one every couple months or so. So human data is not as harder to get. And actually doing it in an actual human anatomy it's almost never been done. It's mostly just excised hearts from transplants. So, but we do work on this. So I've been involved in some of these experiments. Yeah. Yeah. So,
Question.
yeah.
You talk about the comparison between the modeling and
Mhm.
the actual physical results.
Yeah. Mm-hmm.
Can you quantify that, or to put it another way, how close in accuracy does the model have to come to the actual physiological state
Mm-hmm.
in order to be helpful or beneficial?
Yeah, great. That's a great question. And that is something as modelers we always have to be aware of. And my question is going to be that it depends. So it depends on what the clinical application actually is, because sometimes you can get away with understanding mechanisms by just using generic electrophysiology and setting up. So like you can gain a lot of insights into, for example, what happens within the cells. There's a very famous experiment, it's called the dog bone experiment, and it's literally just in a slab, and they can make the tissue anisotropic, like in cardiac tissue, or non-isotropic, and they just applied a stimulus. So it's a very simplified setup. It's not personalized, it's just very generic, but they actually were able to prove how this like dog bone shape arises from anisotropic tissue versus non-isotropic, and they weren't able to replicate it in experiments until I think like 10, 15 years later. And so my answer to this is that it depends, because you can gain very meaningful insights without having a fancy model with lots of parameters and have it fully parameterized. But in some cases that is needed. So if you really want to get patient diagnostics and understand, okay, where do we ablate in this patient? It has to be personalized enough and complicated enough and match the reality of what is happening in that patient in terms of the electricity and this clinical comparison to be able to drive meaningful insights and also in different clinical applications. So I think it just depends on what the application is and how, I would say also how close to the clinic it is. So you can derive mechanisms, but if you're if you're doing clinical applications and using it for patient care, which has been done, then it better be realistic and have a full comparison. But all of this is tested before this is ever used in the clinic. So, yeah.
I find the sex-specific population very fascinating because
Yes.
I think heart is one of the organs that shows a massive difference between
Yes.
males and females in terms of anatomy and function.
Yep.
And I wonder, in terms of variance, let's say you have you construct models to explain or predict whether a certain set of features of the heart signals underlie Wolff, Parkinson, what syndromes or some others. So there's some certain amount of variance between those two in terms of the signal. And what's in comparison the amount of variance between the male and female? I'm just curious, what the effect size there is.
Yeah. So there's a lot of differences. So it's known that like males generally have a bigger anatomy. That's just inherently, so they have a bigger heart. So that's just anatomically derived. There's also differences in terms of, because they have smaller anatomies, they also have different conductive properties. So women will generally have a faster, this arises in the ECG, they'll have it's a shorter QRS complex, or a QRS duration it's termed, and that's how fast your ventricles contract. They'll generally contract faster. I'm using that as an example because that has huge clinical impacts about whether they receive devices or not, and whether they do that. There's also differences like down to the cells, and there's also differences on these hormone cycling about how and how you do the cells represent in different parts of the hormone cycle. Also with menopause, with aging, this will change how hearts and this leads to different manifestations of different diseases in men and women, and making you more susceptible to certain, how you respond to drugs, how you respond to certain treatments. So there's a lot of differences. The thing is, the variation, and why I haven't answered, I guess, the question specifically, is the variation is not well understood. And that's something I want to use the models to understand. They don't necessarily understand a lot of cases, like going through all the clinical guidelines. how are there differences between men and women? They've done this for one application, which is this Q restoration. It's shown Q restoration is shorter, and they've updated the clinical guidelines. But this hasn't been done for the majority of the guidelines. And so they actually don't understand, and we don't understand the variation in how this changes over hormone cycles and age and between men and women. So
It'd be fascinating to train the model on male data.
Yep.
And then actually apply, you have the true labels for female data and show how inaccurate it can get.
Mm-hmm.
And that would be really convincing. And
Shines.
people, you guys have to pay attention to this.
Yeah, yeah. I think, luckily for me is that it is something that everybody's kind of aware of. Maybe they're not doing it, but they're definitely aware. So every single cardiologist that I talk to about this, they're like, oh, I want to understand that. I know it makes a difference, but I don't know why. They don't understand how to update the guidelines. And so that's something that I want to pursue, and that's something I'm really passionate about. And it's something I've been asking, I think, since day one. I've been asking, why aren't breasts integrated into these models? And they're like, oh, it just hasn't been done. And a lot of the experimental data is biased. And so we always make sure we have females in our cohorts, and then I can make comparisons. So this is something I want to answer, and I definitely want to use modeling for. And also machine learning. I think there's a lot of aspects. Yeah.
As a follow-up question to that, you mentioned that there were differences in the cells.
Yeah.
What differences do we know, or what's known and what's unknown about the differences between the cell populations between the male and females?
Yeah, so I'm honestly like that. Cellular electrophysiology isn't necessarily what I've focused on, so I can't answer that right away, and I wouldn't be able to know all the different ion channels and gating, and everything that goes into that. That is something that I definitely want to move into and have started to look up and done the research for, but I don't have an exact answer right away. I've been looking into a lot of how the models are set up and what are the animal models that are used and how that is comparison to humans, and how when they construct these cellular models, when they're used, how much of that data is built on male and female. And these are differences. And luckily for me, because I'm kind of moving into this domain, there are a group, it's called the O'Hara model, and they've set up a sex-specific models. So they have a female version and a male version, and they got actually cohorts of animal models, and were able to set up a sex-specific model and a male model. I have not yet had the time to go through that explicitly, but I've started integrating it into our models. So I have the code, I just haven't done it yet.
So for the models that you're developing, which parameters for the sex differences are you putting in? Is it largely just the anatomical differences that are going into your models to start?
question.
Or?
Yeah, so I'm starting in the cellular electrophysiology. I'm going to go and integrate the cellular model that has male and female, and I have to better understand what the variation is. I don't think that's well understood at this point. So cellular electrophysiology, so the actual cellular model that goes into at different parts in the heart. Another aspect is His-Purkinje systems, which is this kind of coordinated, it's like Google Fiber in your heart. You have a very fast pathway of different fast conducting cells in your heart that allow it to contract very quickly. It is not well understood. This is one parameter that gets encoded, and this is a main point of personalization in the models, and that's not well understood. So they don't understand even the basic structure. It's really hard to measure experimentally, and also clinically, it's not well understood in humans or in animal models. And so the variation in men and women, it's not well understood, but that's one parameter that I definitely want to look into, because this has huge clinical impacts and how we set up the model. That's a major parameter I want to look into. I also want to look into conduction velocities. So conduction velocity, or how fast your heart propagates electricity, and the anisotropy in that is something that can also have huge clinical impacts about different various metrics. So conduction velocity, so how fast electricity propagates within the ventricles and the atria, the His-Purkinje system, different anatomical structures, and how they vary between men and women. So it's not just the anatomy, it's literally the electrophysiology itself. Anatomy has been more explored because it's not a modeling problem, it's a segmentation problem. And you can do comparisons and build shape models and understand variation in anatomy if you have a big enough cohort. The electrophysiology is what's not well understood, and that's what I want to explore.
Related to that, there
Mm-hmm.
probably a lot of diversity. Related to that, there's probably a lot of diversity and specifics of ion channels that, we know some, of course,
Yep.
to disease.
Yep. Yep.
how would you incorporate that in, other than the really big ones, how would you incorporate that into a bi-domain approach?
So the biodomain, so the cellular model gets fed into the biodomain. Obviously, you have to represent your ionic channel in, oh, no, you're good. You're good. So the cellular model gets integrated into the biodomain. It gets integrated into the representation of how the membrane voltages are generated in the biodomain formulation. And then you have a cellular model which generates this action potential and how the cell propagates. And this is how when different ion channels and channels turn on and off. And so by just having a representation of different parameters in the ion model, we can then directly integrate that into the biodomain formulation. Does that answer your question?
Kind
Okay. I mean, I have a slide that shows the.
There's a tri-domain and quad domain and.
We're not doing that. I'm sticking biodomain. I mean, also, if you think of it just explicitly as it's kind of a problem of like, let's say, I'll go back to a really simple analogy. When you have a wave in an audience, right? So I like to think of cardiac cells as kind of a wave, and you start with the person who raises their hand, and then the person next to it raises their hand, and person next to it, you can say how each person raises their hand. So we have a model for how each person raises their hand. And the biodomain formulation does how fast the wave actually goes around the circle and at what speed. But the wave at which you raise your hand, like, I might raise it like this and another person might raise it like this. This is defined by the ion channels and the actual cellular model. And it gets integrated into the biodomain. So we have full control over all of the different parameters. I use already established electrophysiological models for doing this. But yeah, that's the general idea. Yeah?
Yeah, so whether you're talking about,
Yeah.
whether you're talking about updating the ion channel models
Mhm.
or
Gating
the
parameters.
anatomy of the His-Purkinje system.
Yep.
Details, these are at least primarily experimental questions, right? Or
Yes.
How,
A lot of it
how
is,
are, how are
yes.
how do these things exist?
Mhm.
And then you want to work them into your models to better understand patient-specific outcomes and so forth.
Yes.
That all makes perfect sense, but you need collaborators in the experimental world, and you said at your talk on Friday,
Mhm.
I think Rob asked you a question about, I'm not sure how you phrased the question, Rob, it was weird, it seemed like a cardiac insider question. You did, about collaborations,
Yeah,
like working
yeah.
together in Europe versus working together in the US.
Yeah,
And you
yeah.
went to the Europe side real good,
Mhm.
how
The
much
US side.
difficult, yeah, US side.
US side, that's a really great question. So here, a lot of my work has actually been done here at the ski with Rob and Ravi Ranjan and also Derek. So I've integrated with those groups. So a lot of my experimental efforts or this validation effort and all of this, I don't do it myself, but I collaborate with their groups who do very invasive studies and they're very good at it. And I reuse that data. So that's kind of like one form of collaboration that I use to do experimental validation. Outside of that, in the US, I have a lot of interactions with Linwei Wang, and she's in Rochester, New York. She does machine learning, and this is another, I guess, this isn't validation, but this goes into machine learning and using the models to do pace mapping in different clinical applications. And then I'm also affiliated with Pat Boyle. He's in the University of Washington. And he has a group here, and he does very similar mechanisms, but he has kind of a different approach in a different direction. So he does a lot more atrial modeling. So that's kind of my US collaboration and group. The experimental, I guess if I get back to the experimental validation, so Rob and his group and their data, and also Ravi Ranjan, and they have experiments. I also work with a group in Bordeaux, France. Jake goes and visit them periodically. So Jake Bergquist, and he does the experiments there. And that's with Laura Bear, and they do very invasive either optical mapping or these invasive torso tank experiments like what is done here. And I usually work with these groups and collaborate with them to get this type of data if I'm doing model validation. But a lot of that is, yeah, hopefully I can build that collaboration here stronger at the University of Utah.
Sure.
Mhm.
And how do you imagine that, those collaborations supporting your fundraising efforts going forward? Are they gonna be supporting your proposals?
Mhm. Yeah, so everyone that I'm collaborating with, I've had really open and honest conversations about grants and moving forward and what I want to pursue and what I want to do. And they've all said, oh, I'm happy to contribute data into this and I'm already doing this. And it's kind of like, for example, with the data with Rob or with the data with Laura Bear, it can be an R21 reuse, a reuse of scientific data that was meant for something else and moving forward. And in terms of like the modeling workflows and all these tools, they said, yes, you can definitely move forward and we support you and we'll help you. You can craft this and you have full access to all the tools. So every single person that I work with in my collaborative network has been fully supportive and willing to be a part of kind of my journey forward into independence. So it's been fun. Yeah.
So my question is kind of along the lines of what Chuck and Orly asked, which is like,
Yeah.
if you're reusing this data and you have
Yes.
collaborations, right? You're using Rob's data,
Yes.
but you want to study what happens to heart conduction velocity during childbearing years versus like menopausal years. You're
Yes.
relying on Rob having collected that invasive data.
Mm-hmm.
So,
Yeah.
like, how will you tackle, which I think is really cool, really important work, but how will you tackle that when relying on collaborators to have collected those very nuanced, sex-specific animal
Data.
models? And
Yeah.
how
That's a great
often
question.
do people keep dogs and mice
Yeah.
into postmenopause, right? So
Yeah,
it's
yeah.
really specific, and it's hard to reuse data like that.
Yeah. Honestly, there's a lot of data that just hasn't been done. So in terms of experimental data, and especially moving into sex-specific, I use it for model validation to understand if I have the baseline proper and if I want to explore different sex-specific mechanisms moving forward. I would either probably have to start that group myself, but I've always stemmed more on the modeling side and the mechanistic side. So I don't necessarily need experimental data. Then I would move into clinical data and clinical collaboration. So as long as I have access to the clinical data, which is very invasive, so they have clinical mapping system data, for example, gathered here at the University of Utah and from our clinic, I can actually revert to that type of clinical comparison on how, from endocardial mapping data from the clinic and different diseases and different disease states, from menopausal women, from aging men, from these different groups, which are all accounted for in these different biobanks. And so I would move forward into that, understanding the basic mechanisms from experimentation. That would be honestly something that would be hard for me because I'm not an experimentalist and I'm not going to pretend to be. But I can use these models and set the parameters and say, okay, based on previous work that has done, maybe with someone I'm not even collaborating with, I can start to parameterize the models based on clinical data and start to bring it all together and being able to still explore this domain. There's so much to be done on that. So that would be my backup question. Yeah?
Yeah, and I had a follow-up question.
Yeah.
That is, I don't know anything about the cardiac data
Mhm.
side, but I can imagine there are billions and billions of ECGs
Yes.
collected worldwide.
Mhm.
What is the current state of open data and open science in this kind of framework?
World. Great question. I think there is a lot of idea of open science and open data across this field. In terms of, let's say, I'll start with databases and I'll talk about software and modeling. So databases, there are a lot of public databases of ECGs that are available. So there's P2B Excel that's in Europe. There's also the UK Biobank. The UK Biobank is a tricky one because you actually have to pay about £3,000 per year to be in the club, which can be paid for by grants and other fees. And so there are large increasing databases. There's data here. So I was talking to Ravi Ranjan about available clinical data in terms of ECGs. I think what's important for my work is not just databases of ECGs. I think that's thinking of it in terms of machine learning and diagnostics. I need large databases that have MRI or CT, some sort of anatomy, plus ECG, and it needs to be standardized. And the only database that I've seen that, not even the database at Mayo Clinic, for example, their ECG database, is the UK Biobank. So you can generate massive amounts of data, which is both anatomy and electrophysiology, which is what I need for mine. And that just hasn't been paralleled in terms of the UK Biobank. And they're working on here at the clinic. So Ravi said they have a lot of data of different disciplines and MRIs and CTs and ECGs. But I would say in terms of the open science sense, they're generally not yet to the state which you're working just with smaller cohorts, so, under 100 patients. And if they are open science, they're open available, you usually have to pay for them. But they are technically open. And then in terms of the workflow and the workflow tools and all of this, I think, as I said in my talk, it's still, we're pushing for open science, and this is very important in the field in terms of reproducibility and being able to check each other's work and making sure that the modeling aspects are valid. And I've seen so many PhDs do same work that I established, and it's kind of a shame to see somebody spending so much time re-implementing the wheel. And so there's been a lot of work to make all of the tools open science and available and publicly available. All of my data goes on Zenodo. There's open GitHub. We manage a GitHub repository. We're placing all this code. We have an open modeling initiative. It's like OpenCarp simulation framework that we integrate into. And it's all very open and nice. We like to think that, but a lot of the tools that you need to really make a lot of impact in this field are still closed source, and they will stay that way until they were been fully exhausted. So, and what I mean by that is like my His-Purkinje code that I've generated with these physiological detailed meshes, they're not publicly available. The UVCs that I generated, which are these cardiac coordinates, they were not made publicly available. And different aspects of the simulation framework are not openly available because we're still leveraging the science that comes from that. But we're definitely working in that direction. So I think because it's such a collaborative field, there is a lot of open science and work and collaborating of modeling and data and all of this. But it's, from what I've seen, it's generally within collaborations that have scientific profit on both sides. So, yeah.
Just because you mentioned the UK biobanks,
Yeah.
I know they also have a lot of genomic data in there.
Mhm.
I'm just curious if it overlaps with the same patients where they have ECG and MRI for, so.
Yeah, they do. So actually, one of my collaborators, he's in London, so I'm actually studying in London right now. They're going to start leveraging that. So because they have databases which are Cine MRIs, and they have an automated way for generating anatomy. So they have anatomy, which is what you need for modeling. They have the ECG and other forms of electrical data, which is the EP or electrophysiology data. And then they have all these other parameters of patients, patient factors, whether they have diabetes or not, genomics, and they're starting to pair it together. And it's literally just like it's like a data mine right now. And they can leverage it because you can generate all these models and then compare it to patient factors and how they represent themselves. And that's currently what I'm in London doing and learning how to do and leverage this type of like population data and draw different factors and how that changes in diseases. So
Really awesome. So just, I'm curious, so how many patients are there that have all this data?
Oh, I think the latest number they told me was 60,000. 60,000 patients I think they've collected up to this point. They've only built models for about 5,000. 5,300, and they're not open source because you're not allowed to share unless you've paid this £3,000 a year. But they're going to make an average database. They've averaged this 5,000 and they're making it publicly available, which is going to be fun. But it's 60,000, I think, at that point. So they're a huge, it's just like a fun hotspot for machine learning and population modeling. Yep.
Jessica's question.
Yeah.
The invasiveness and non-invasiveness, how much more information do you gain if you actually put needle electrodes inside and out? I understand the model.
Great question.
Production much better. But is the ECG perhaps good enough to at least classify disease state, or do you get 10 times more information if you put needles in and I guess 10 times better?
I would say when you put in the needles, they put in about 30 needles and they have about 10 electrodes each. You can assume that a lot of those will be injected into the blood pool. So say you lose 2/3 of those. So you're looking at 20 needles each with 10 each. And then you lose a few during registration. When you actually, they take an MRI and they'll actually lose the location of the needles, and then they have to throw that data out. And so you get a lot of information within just the LV. So the needles are always injected into or like inserted into the left ventricle of the heart. They're not inserted into other parts of the heart because the wall's too thin. So you get electrical data in a very specific area of the heart that's concentrated. They call it the left ventricular manifold. And you only get data in this very specific area. If you can think about it, the heart is very heterogeneous. And if you're only looking at a little part of the LV, you're getting a very kind of like closed look on what's happening. But the nice thing about this data is that it is volumetric. So every other type of clinical data, most experiments, it's very much just on the epicardium of the heart, which is the outside of the heart, or it's on the inside of the heart, the endocardium. And there's not a lot of information about what is actually going on within the heart, so within the wall, within the volume of the heart. It's not understood what is happening during ischemia. How does it diffuse? How does it spread? How does it grow as the tissue starts to die? This is what a lot of their work has focused on. It's not understood even how different cells vary throughout the wall and how you have different representations of tissues and cellular electrophysiology. And these signals, because they're EGMs and you can construct unipolar or bipolar electrodes, you get a lot more of information about what is happening in terms of anisotropy, conduction velocity, these factors. And in a project that I have right now with Rob, we're working on developing or personalizing or parameterizing this model to match the data so that we can gain even more insights into the electrophysiology and conduction velocity from this invasive data. So I would say, to answer your question, it is limited in scope, but what it gives us in comparison to standard experimental techniques or clinical techniques, it's very valuable. Yeah.
What's the state of two-photon optical mapping?
Oh yeah, that's a question. So a group in Bordeaux does this, and I've worked a lot with them on terms of optical mapping, and they do iterations of testing out different techniques with this optical mapping. Optical mapping has the same problems, at least in the experiments that they do, that it's still on either the endocardium or the epicardium. Because it's optical mapping, you get what's happening on just the outside surface of the heart or the inside surface of the heart. And what?
You can't get very deep with multi-photon?
No, I don't think so. I don't think so. I've never seen anyone do, like when they do slices through the heart or anything, you can't do that because then you lose the electrical function. And so it's literally they take your heart and they cut, like here's a little heart, they'll do a cut here and a cut here, and they'll place it on this like square block and kind of pull it, and then they can see what's happening in terms of arrhythmias in a flat manifold, which can be very debatable in terms of how you're disturbing the electrophysiology from a normal heart beating in a chest. I've only seen optical mapping ever be applied on the inside or the outside of the heart and not volumetrically, because you lose the function once you start to cut the heart. If you stick electrodes in, you're doing pretty good. You still lose some function, but yeah. So that's what I've seen. Anything else? No? I have grand ideas. You guys want to hear grand ideas? Yeah? Okay. Oh, this is my research network. We talked about this. All over Italy, Utah, Pat Boyle in Washington, the main PIs that I work with in Austria, in United Kingdom, who I'm currently studying with, Steve Nutter. It's kind of the overview. I found this really cool tool. It's called VOZ Viewer. It takes your publication record and it will show you all your links and makes an interconnected map. That's what this is. If you guys want to try it out, it's really fun. Anyway, so collaborative grant ideas. So these are the grant ideas I have moving forward. So these are 5 of the different grants that I've kind of thought of that I want to pursue. They're all in different directions with different timelines and different amounts of background. The first one is XHeart, which is the sex-specific virtual heart technologies of electrophysiology. This would be a repurposing of the grant that I already just got submitted, but I want to update it with cellular electrophysiology. So that was one of the main, that was like the only negative feedback I got from the revisions. And I want to integrate that in. I have Platinum, I like coming up with names, Platform for in silico drug testing and cardiac models of electrophysiology. I also have Interactive EP Platform. This is a training tool. Then I have BPERKY. This is an interesting name, but understanding the behavior of the His-Purkinje system during ischemia, and then also ACE, which is AA-powered cardiac digital twins of electrophysiology. So which one do you want to hear? What are we? My first publication. my first application, I want to be this X-Heart because I've already finished it. It's a 12-page application, and I just want to build on it and focus more on collaborations here. So that's what this is. I already submitted this to the Austrian funding mechanisms here, and I got submitted and funded. But based on their feedback on how I've kind of started to work with this project, I would want to expand on it a little bit. But I have the proposal generally written. What this is this is kind of the starting point for my sex-specific modeling, population modeling endeavors. I have 3 basic aims. The first is to generate populations of male and female cohorts. This would be predominantly based on the UK Biobank data. That's what I'm doing right now in London, so I will have preliminary work. The second would be generate a cardiac digital twin cohort. So I've also made a lot of groundwork on generating personalized patient-specific models, so from male and female, and I will be able to compare the electrophysiology after they've been personalized in the 2. And then the third aim would be to validate the sex-specific models, and this would be more of an analytic in terms of feature distributions of if I have a population, does it match the distribution of Q restoration in different diseases? Does it match the population of these different morphological characteristics? And then I can start to understand that. And then I also would apply it to a disease called bundle branch block, which is where part of your heart is not functioning or activating as fast as it should, and they do resynchronization therapies. So I want to apply it to that to understand what the predictive capacities are for these models. Kind of the funding mechanisms that I've designated are just an R01 with the early stage investigator bonus, the NSF Career Award, or the AHA, the American Heart Association Career Development Award. And this, I've kind of done it in Austria as a kind of a starting project where I work with collaborations in terms of clinical data and also with cardiologists, so clinical cardiac electrophysiologists, because they help kind of guide to make sure that the project is clinically applicable. But I'm also looking for, I would need radiologists who can do the imaging if I want to generate new data. And because I have very specific torso models that I like to work with that are ideal for this project. I have the 14 I already gathered, but it would be great to start making this more routine. And then I would also want experts in shape modeling, uncertainty quantification, population modeling, and machine learning. So I've kind of, I've already started working with Akil on doing uncertainty quantification and starting a project with him and how that integrates into this project. I really want to work with Shireen and then also Sarang in terms of machine learning and maybe other approaches. So that's the first grant idea. I've thought about this and written it quite extensively, and I would want to reapply it here in the US pretty soon. Yeah?
I think this really has a huge potential for career award by the NSF.
Mm-hmm.
So one of the components is incorporate teaching into the research. Have you given that some thought? So.
Yeah, exactly. So this is my other grant, and they pair together. So can I go to the other grant maybe a little bit and show what my ideas are in terms of that? So let's go to this one. Okay, play from current slide. So my other grant idea, and this is more kind of like industrial development, is I want to, oh, it's not showing. On my screen it's showing as I'm presenting, but on this one it's not. I don't know how to, oh, there it goes. Okay. So I am actually working, we've done extensively in our modeling framework and our modeling setup and our workflow to generate, we have a tool, which is kind of an interface that you see here, that we can set up simulations and understand you can change all of the parameters in the electrophysiological model on the fly. You can change conduction velocity, run a simulation, see what it looks like. You can add pacing sites, you can put everything, it's completely interactive and how you work in, and then you can generate signals. You can start to analyze this data. We have integrated clinical mapping data and devices and electrodes and how this all comes together. And I'm really pushing to generate this as a teaching tool, not just for medicine, but also for students who want to learn cardiac modeling and electrophysiology. And we've already have this working version. This is the full scientific version. I want to repurpose it for a training tool. And once I have this, then I want to integrate in this sex-specific modeling into training and education and we take this and we actually work with cardiologists and we show them demonstrations of what is happening in the heart. I can show them the mechanisms of what's behind the signals, how this comes together, how this interacts with the imaging data, what are their registration errors, and you can start to derive all of this stuff. So my plan is to develop this as a teaching platform, so take the big version and make it kind of a smaller, more confined version, which can be used for educational purposes. And then this is a platform to serve and show these sex-specific differences and really show the mechanisms about why this is happening and why this goes into clinical treatment. So that's my plan, and this is one of the second grants that I want to apply and work with, and maybe work with industrial partnerships. The company that actually develops this is on board, and I've talked to them, and they said that would be great. We've been thinking about it for years, and so they would be completely viable. So there is this NIH one with BPI that I was looking at, so that would be one aspect. And that's kind of like the teaching platform, and I want to integrate everything I'm doing into this so we can actually go and show how it works. And this is one of the benefits of mechanistic modeling, is you can say, hey, here is what is happening in the heart. This is what the influence is on the ECG, and you can actually just show this in a system that's very easy to understand.
So I think if you took this very powerful and somewhat sophisticated tool and simplify that so that, you know, people and pupils can use that and get excited about heart function and differences, then I think that's essentially funneled by the answer.
Yeah, this takes away all of the basic mechanisms of the hard part of cardiac modeling. So, you know, it took me 5 years to get trained. Now we have a student who's running the same simulations that I am in 2 weeks because he has this tool and he can just change and update all the parameters. Maybe he doesn't understand exactly what that means, and that's why we've developed other parameters to make sure that there's no simulation artifacts or problems that come in from inexperienced users, but this tool is exactly what we want to do. And I want to make it a smaller version and then resubmit it. So that's my plan. So other grants, we can maybe go back to the beginning. So we've talked about the interactive EP platform. Is there any more questions on this? Oh, it's okay. It's taken its sweet time. Here we go. And XHeart, I also have 3 other grants. Anyone? Come on, I want.
So I have a question about the platform. Is that
Yes.
going to be open source? By which I mean, so that students could just not use this for training and just running the existing code, but maybe could also add code modules of their own.
Yeah, so we talked about this. We were thinking about if any of you are aware, there's a tool called ECG Sim, which in its essence was a very similar tool. It was developed in the Netherlands from a professor called Peter van Dam. And I actually learned that here at the University of Utah in the bioelectricity class. And we want to take this tool and make it so it's open source. Everybody can have access to it. Everyone could use. I think there would be some discussion about whether or not the source code is openly available or whether or not it's just a shipped binary that's downloadable, because once you open up the source code, that opens up other issues of things. So this is still a discussion. We're still having it. It depends on what form that it would take moving forward, but it's definitely a discussion. So a lot of the basis of this tool is actually already open source. So the basic mechanisms, it's just certain aspects that are still contained. But yeah, so that would be the plan. I want to make it as widespread as possible and encode it with different diseases and also have some flexibility. Yeah. Yep. Digital twins, which one is that? The AI powered one? ACE. So I'm very happy given my conversation with Sarang this morning. I came into his office and he was like, Karli, digital twins and machine learning. And I said, yes, please. So I would like to take all of these different aspects of cardiac modeling, which can just be redone with machine learning at a much faster rate because we have the data, we have the possibility to generate massive amounts of data. And I want to go through really step for step and replace all these different aspects with machine learning and generate essentially AI-powered cardiac digital twins. So, and what I mean by that is I want to generate a neural network of different cellular models and ionic models. Because you have the capability to do this regression of parameters to output in different morphological aspects. We've done this in one of my other projects. I want to generate a surrogate model. So this has been done in some capacity, but these surrogate models just run unparalleled. They have 1 second of computation time versus 8 to 10 to 20 minutes to 24 hours, and you can just generate the surrogate model and it runs all your code very efficiently. This would be a really great update for also the learning platform to make it faster and more interactive. And then I also wanted to do AI-powered cohorts of various cardiac arrhythmias. And I definitely want to expand on this in terms of also shape modeling and kind of using machine learning to leverage this whole entire system. So that was one of the grants that I would want to submit. And all of these aspects have kind of done in little pieces, but now they're never together as an entire system. And I know Tolga's doing some of ECG machine learning, Shireen, Sarang as well, and also Akil, because a lot of these generating these models, you need uncertainty quantification in the background to understand what parameters are actually relevant and what do you actually need to train, how much data do you need. This is a lot that uncertainty quantification can give us. So this is what I would like to, one of the project directions I would like to do that is very beneficial for all my other projects. Yes. Oh, I guess that was, yep. So this is my other one. Other aspects? Nope. I was thinking about it.
Yes.
Okay.
Yes.
No, I don't think it is. Let's see. So, let's see. How much time do I have left? Because maybe. Okay, so I do have two other ideas for grants. I'm going to kind of skip over them. One is to do these experimental databases this would be together with Rob. Maybe I can just swim through. And regenerate it to understand what is actually happening in the Purkinje system because it's not well understood. It's very hard to measure, and we can pair modeling with experiments to be able to understand what is actually happening in the Purkinje system. And this is one grant that I would want to submit moving forward, and this would really work off of a lot of the data that Rob has already generated. So I was thinking the second analysis of existing data sets, R21 as a major grant, and maybe a Trailblazer Award for an R21, so kind of these shorter grants. And this is together with Jake, Tomas, Robin, Linway, which are all collaborators of mine. And then I have another grant, but I won't get into that because I thought it might be good to maybe talk about resources and teaching and things that I would need. So in order to do this research, the main applications that I would need is different computing resources. So I need workstations, laptops, and data storage, and also high performance computing, which is all mostly offered here at the Ski Institute. And then I need workstations, which are about 9,000 per machine. But apart from that, I don't have experimentation or lab space. I just need funding for essentially computation power, and then also software and different databases. And I need access to very specific clinical data. So I need large clinical databases of ECG and mapping system data with anatomy, as I said, specific data set with torus on high resolution cardiac MRI. I have that. I've talked about getting access to it. It seems like it would be pretty straightforward here at the U of U. It just needs IRB approval, but the infrastructure is already here with cardiologists. And the experimental animal data, which I have access to through Rob and Ravi and Derek Dosdell at the CVRTI. Kind of what I see for myself is I would have like a smaller initial cohort of students, and I'd go to a more maximum size. This would be the biggest size that I would want to have moving forward. And in terms of support, I would always need a computational lab manager or senior scientist that's interacting with the group in some way to kind of manage the code and the software basis and the simulations. And we have one in our group now. And because I have a lot of ambitions in terms of developing this platform, I would need a software developer and then also IT support for all the different software and tools. So these are kind of what I would need for my lab to get started here at the University of Utah. Institutional support, maybe if we're I would just want access to grant training workshops, course supervision for the Center for Teaching Excellence in the first few rounds so that I can make sure that my teaching meets the quality of the university, which I expect that it will, but it would be fun to get feedback on things that I can improve on. And I would really like mentors and experience with machine learning and population modeling, larger scale US grant writing, and industrial involvements or startups, because I have a lot of industrial ideas about what I want to move forward and do. And it would be nice to have someone here who has kind of treaded the domain of academia and industry and kind of knows how to integrate with that. Areas of service, I would really like to do a biobank initiative for the University of Utah and get this more going like the UK Biobank. I would like to serve on the committees of the Curriculum Committee and the BME Equity, Diversity, Inclusion Committee. And I would also like to get involved in the student organizations of the Society of Women Engineers and Graduate Women. Let's see, oh yeah. And what can I bring to a classroom? So this is more on teaching. And how I plan to move forward in teaching. So I think what I can really bring to the classroom is I can bring a complete understanding of many interdisciplinary systems and subjects. So mathematics, physics, modeling, coding, and I can bring all of these things together. And this makes it very easy to come up with various real world examples. I've used a lot because it's very nice to see how what you're learning relates to what you're actually doing and how you interact with society. So I have a bunch doing yoga for forces and rapids for cardiac ablation. Students generally understand these real world examples really, really well. I also can bring interaction with clinical, academic, and industrial partners. So I have a lot of collaborators that I can bring into the classroom. And I've gotten a lot of experience on inclusivity of students with different learning styles and backgrounds. So in Graz, I teach in German. I get students from all around the world who are from medicine backgrounds, and my students come from mathematics and computer science and biomechanical engineering and mechanical engineering and electrical. So I've gotten very good at working with students from different backgrounds and styles and different places. And then I think another thing I bring is a drive to learn and improve my own teaching empowerment. So I've had so much fun with this in the last 2 years. I've gone and I've read podcasts and I've read books, and I've been going and seeing how I can think about a classroom and how I can do small changes to integrate this to make my teaching better. And then I guess the last one is curriculum courses. These are the list of all the courses. This is what I'd be enthusiastic to teach. So I'm very enthusiastic about thesis writing and communication. So scientific communication has become very important to me. I'd like to get integrated with those. I can obviously teach a lot of the elective bioengineering classes that are in the cardiovascular track. Computational methods would also be fun for me because I've come from that, and I took that class and I've seen how computation can be integrated into the bioengineering curriculum. And then I also have 2 proposed courses for what I would want to do, which would be like novel technologies. This would use this ECG platform, and I can integrate it to teach students how to think in clinical workflows and what are the clinical tools and what are the devices that are being used, and then integrate that with cardiac and clinical workflows. So that would leverage that. And I have another course I'd like to teach on uncertainty in biomedical systems. So how do we think about uncertainty in terms of biomedical applications and systems, and how can we think of that as engineers and as modelers? And because in every single aspect, even as an engineer, you have uncertainty in your designs, and what are the tolerances and how does this feed in together? And I want to really have a class like this, because when I started modeling, I had no comprehension of what this was and what this meant, other than you have design criteria and what's the tolerance. So I'm very passionate about kind of spreading this, because I think quantifying uncertainty in modeling systems, whether it's any different, cardiac modeling or mechanical modeling or all these, it's always a question. It's always how do you quantify your uncertainty, and how does that affect your modeling? So those are my courses. Do we have more?
Go back, half a slide.
Half a slide?
I just want to see what you can teach that you're not enthusiastic about.
Okay, so there's careers. I mean, I could teach it. I would love to. I think I have a lot of really good collaborations, and I know a lot of friends who are in industry, and I think I would do very well, but it's one of those classes that, I mean, it's not the most fun. So if I have to do a core class, it's like I would much rather, actually, Chuck's gonna die when I say this, but take on biosystems than take on careers.
Yeah, no.
Is it your course? So you're the one who took it over. We can become a perfecta. I'll teach biosignals. You can teach biosystems. We'll work together. I think it's just because it's not that I wouldn't, they're just more close and dear to my heart. So obviously fundamentals of bioengineering too. So this is what Rob and Doug Christensen are teaching right now. That was one of the classes that really made a huge difference for me in deciding to do cardiovascular research. And I'd really like to continue that. So these classes are the ones that I took out biosystems, but I would like to teach that one. Those are the ones that really changed my idea of bioengineering and the department in general, and they were my favorite classes. And so that's why I chose these as enthusiastic to teach, because they are something I'm passionate about. And then also I think, a lot of the other ones. So bioelectricity of tissues. I think that was one with Robbie. He came in and he did ECGs and how does it interact. And it was my first time thinking about things in that clinical application that then has kind of formed my research. So, yes. Yeah.
Okay, I think we've used up the time and the patience of most people in the room.
Sorry.
Yeah, no, it's no problem.
Okay.
It wasn't patience, it was just time, but thank you, Carly.
Great. Oh, no,
So this is a little parting gift. We were cleaning up around the place in order for the 30th anniversary event, and we found this poster with Karli as first author, showing you things like uncertainty quantification and ECG and skeletal models, and just to give you some idea of the diversity of experience that she brings to her profession.
Oh my gosh, I gave this to you as a blanket. Thank you.
All right, here.
Thank you.
All right, thanks
Thank you
everyone.
for your time.
Thank you.
Okay. Oh, good idea. Thank you.
Much.
Thank you for coming.